My name is Victor Huertas. I'd like to start off by acknowledging the Aboriginal people and Torres Strait Islanders as the first inhabitants of the nation, and acknowledging the traditional custodians of the Australian lands where our staff and students live, learn, and work. Our expectation is that interactions during this seminar are constructive and that all attendees behave with respect and consideration for others. I would also like to ask all audience members listening online to make sure your microphones are muted. At the end of the presentation, you will have an opportunity to ask questions, and you may choose to unmute your microphone at that point or to type your questions in the chat box. So with that being said, I'm pleased to introduce today's presenter, Dr. Kate Quigley. I think it is fair to say that Kate followed quite a convoluted path over the course of her career so far. To contrary to what her name may suggest, she is a fellow Spaniard. She was actually born and raised in southern Spain. And when she finished her high school, she decided to cross the Atlantic Ocean to go do a Bachelor of Science in Biology at the University of Texas in Austin, the US. After she graduated, she moved to Peru, where she served as a Peace Corps volunteer. And after her time in Peru, she decided to cross the ocean again, but this time the Pacific Ocean, to come here to Townsville to do a master's first, which then she followed with a PhD here at JCU under the supervision of Professor Betty Willis and Dr. Lina Pei. After she finished her PhD in 2016, she has been working at Ames, first as a postdoc and later as a research scientist. And just recently, in the last few months, she joined the Mindaroo Foundation, a foundation that she served as the senior research scientist and also as the research director of the Mindaroo Exmouth Research Laboratory. Kate, thank you for joining us today, and please take it away.
Thank you so much, and thank you everybody for joining us today. And I guess in conjunction with the acknowledgement that Victor gave about traditional owners, I'd also like to give special thanks to the Llama Llama traditional owners in the far north of the Great Barrier Reef, where a lot of the samples that I'll be talking about in this presentation, those samples were collected from. So today's presentation will kind of be a dual presentation. So I said yes to this talk when I was still at Ames, so I'll present some of the work that I was doing there. And I chose the work that I thought would be most relevant to the ARC Center of Excellence, given the long history of looking at bleaching and documenting bleaching on the Great Barrier Reef. And then the latter part of the presentation, I'll talk about Mindaroo Foundation, because as I speak to people around Australia about Mindaroo, some people know it and some people don't. So I'd like to introduce you all to some of the work that we do at Mindaroo Foundation. So firstly, I'll be talking about symbiosis restructuring following mass coral bleaching on the Great Barrier Reef. And this work was done as part of a NESP program looking at and responding to mass bleaching after the 2016 and 2017 bleaching events. So coral bleaching on the Great Barrier Reef, many people at the ARC Center of Excellence have been involved in documenting and charting the absolute devastation that's occurred due to bleaching in 2016, 17, 2020, and unfortunately again now this year. And the most concerning thing is that as we document water temperatures year on year out, it seems that on average, water temperatures are increasing, suggesting that bleaching and the manifestation of bleaching will become something that is a pervasive feature potentially on the Great Barrier Reef in the years to come. So what is bleaching? If we take this little diagram here, we can see that on one side we have this brown coloration, and that would be characterized as healthy tissue. So that coloration is due to the tiny dinoflagellate cells that live inside the coral tissue. And those dinoflagellates from Symbiodinaceae give the coral the nutrition that the coral needs to deposit its skeleton and to grow. But when warm waters hit, or when sometimes in conjunction with high light, and very still environments, we get something called bleaching, which is the breakdown of the symbiosis and the loss of those tiny dinoflagellate cells that live inside the coral tissue. And then we can kind of see almost right through that translucent coral tissue, the coral animal itself, and we can see the skeleton underneath. And bleaching doesn't necessarily always lead to death. So if those bleaching conditions stick around for too long, so if high temperatures, still conditions, high light stick around for too long, the coral animal can die. And then that's when you get the overgrowth of this filamentous algae seen on the far side of this figure. So it's really the breakdown of the symbiosis between these tiny dinoflagellates and the coral animal. And so generally, when we think about tropical biodiversity, we think about all the different coral species, we think about different fish species, but we should also be thinking about the dinoflagellates that, the many dinoflagellate species that live inside of corals, as well as free living in the environment, which transmit back and forth between corals. And here we have just a tree with a very simplistic tree of some of the most well-described and well-characterized symbionts. And for this talk, I'll just be focusing in on the generally stress-tolerant or heat-tolerant Durastinium, seen here in red. Definitely not the only stress-tolerant taxa, but generally the ones that people think of when they think of hardy symbionts inside of corals. So we know that climate change is disrupting biological communities, and there's been some really nice papers to show that fish communities are disrupted, corals themselves are disrupted, but we really didn't have an understanding of what was going on with the symbionts inside of corals, as well as free living in the environment. So that's what we set out to understand. So coral, there about 20 years ago, there was a hypothesis called the adaptive bleaching hypothesis, which essentially looked at bleaching like an ecological opportunity and essentially posited the question, could reefs be using bleaching as a way to survive climate change? So say we take our little toy example here of a coral, and there's two different taxa of symbionts that live inside of it. The red cells, which are the heat tolerant cells, and then the orange cells, which are the less stress tolerant or heat tolerant cells. And say maybe through time, we get some bleaching event and corals are able to change the relative proportion of these two members in their community to be one community at the end that's more able to withstand higher temperatures, either in the present or into the future. And this is something called shuffling. So the change in the relative abundance of the community that's there already. Alternatively, another mechanism that corals can use to essentially extend their phenotype is something called switching. And that's where corals can potentially take up new individuals, new taxa from the environment, generally in the water column or in the sediments, and then essentially form a new community where maybe the relative abundance as well as the actual taxonomic composition inside the coral is different. And the whole purpose is that potentially this new community would allow those corals to survive better into the future. So this becomes really important when we think about metapopulation structure. So the Great Barrier Reef is a composite of different reefs, and there's flow in between source and sinks around on the Great Barrier Reef. And so essentially, if we're looking at understanding what's happening with surviving corals, it's really important to understand how do healthy reefs potentially reseed degraded reefs down the line. So that can either be the reseeding of surviving corals, the offspring of surviving corals, or alternatively symbionts that allow the corals to survive under these more challenging conditions. So potentially the reseeding of particular symbionts to degraded areas around different parts of the Great Barrier Reef. So this is why it's really important to understand what's going on in the context of what is surviving after these mass selection events. So in order to answer this question, we needed a long-term data set. And so we were able to go into the tissue stores at AIMS, at the Australian Institute of Marine Science, and collect tissue from 2003 up until 2016. And then we supplemented these historical tissue collections with new collections on the AIMS boats or in conjunction with the Great Barrier Reef Legacy. And we ended up being able to put together a data set of 26 different reefs, both in the North and the Central Great Barrier Reef, where the bleaching and the mortality was concentrated in those years. And we could divide the reefs into pre-2016 bleaching and post-2016 bleaching. And so we took those tissue samples, about 1,400, and we selected 3 species in particular, 3 species of different acroporids. And we were looking at species that had different bleaching sensitivities. So we wanted to see, could the symbiont communities be driving any kind of differential survival in these 3 species? In particular, Acropora tenuis, which is generally thought to be a pretty tough species, a pretty hardy species. Acropora millipora, the kind of lab rat of corals, and then Acropora hyacinthus, which is generally thought to be a pretty sensitive species. So we had historical tissue samples, new tissue samples from 26 reefs of these 3 species. And then we also had sediment samples that we collected on our new voyages, as well as sediment samples that I had collected and analyzed during my PhD. So we were able to have a time series of both before and after bleaching across these 3 species, as well as free living in the environment. So if you think coral taxonomy is a nightmare, their symbionts aren't any better. And a lot of the challenge in determining what symbionts are there is because of the difficulty in describing intragenomic versus intergenomic variation, where that really makes it almost, well, it makes it very challenging to even say, well, this is a species and this is not. And so what you have to do is you have to select a marker to use, and generally the ITS2 marker is what we go with. Unfortunately, many of the markers for Symbiontaceae are multi-copy, which means that within the same genome, there's multiple copies, even within the same genome, within the same cell of this same marker. And to kind of make it even more challenging, these different multi-copy variants have different variation within their sequences. So essentially that means that even if you had one cell, it still can make it slightly challenging to describe, you know, is this a species or is this not a species? And so there's really at the moment, two main ways that you can characterize symbiont diversity, either through something called SymPortal or something called the Data2 pipeline. And neither is wrong or right, but it is important to note that what you use is dependent upon what your biological question is. So for example, if you're looking at more high-level questions across populations, SymPortal might be okay, but you're going to lose a lot of potential interesting diversity on the reef scale or the individual scale, compared to Data2, which generally you could get more false positives, but you're able to really drill down into that nitty-gritty of diversity if you're looking for small scale changes. So those are the 2 options that you have at the moment. And so if we just kind of go into some of our results, we were first interested in just asking the really basic question, where are the heat tolerant symbionts? So looking at prevalence. So prevalence can be described as just presence or absence. So if we take this little example here, we can see that Coral 1 and Coral 3 each have these heat tolerant symbionts, but Coral 2 does not. And so when we looked at all of our corals across our different reefs, across our different species, we could see that for this heat tolerant symbiont taxa, before 2016, most corals did not have these the signature of the heat tolerant taxa. And it was only after the 2016 bleaching event did one of the coral species, Acropora millipora, which is generally thought to be a more flexible species, did it actually show an increase. So more colonies had these heat tolerant symbionts after bleaching. And the thing to note here is that there was quite a lot of variation for these 3 different species, which was slightly unexpected. And then when we looked at our symbionts, we actually found heat tolerant symbionts in almost every sample. So about 99% of our samples. So the heat tolerant symbionts were there, but it wasn't until after 2016 that particular coral species, specific coral species, were actually able to take advantage of them being in the environment. So we were also interested in looking at the diversity. because within Durastinium, there's different types of Durastinium, different species of Durastinium. So again, we looked across our 3 species, we looked in our sediments, and we could find that there were hundreds of different punitive taxa within each of these areas. And in particular, in the sediments, we found much higher diversity compared to the corals. But interestingly, there was about a 31% overlap between the taxa that were found inside of corals and in the sediments. So this again suggests that this transfer, this linkage between these 2 biomes after bleaching could potentially be important in terms of this idea of an ecological opportunity presented by bleaching conditions. So now looking at diversity, where each of these different points represent a different taxa within that heat-tolerant Durastinium, we could see that more or less the diversity was pretty consistent within corals. But interestingly, after 2016, we found an explosion of diversity in this heat-tolerant group. So that was really interesting to see that, you know, even though they were more or less within every sample, something was definitely driving the explosion of their diversity after this first bleaching event. And when we looked at diversity across all of our symbiont taxa, so now not just within Durastinium, but across everything and by region, so in the north versus the central Great Barrier Reef, we saw that in corals overall, there was a decrease in diversity in the north, some varying patterns in the central region, but that again, we saw an increase in diversity across all of our taxa in the north and in the central region. And probably most concerning is in this northern area where we really should be asking, okay, we're getting much more diversity, but they're in the sediments, but we're losing diversity potentially in some key coral species. So something to note. So we were seeing these kind of different ecological patterns in our symbiont communities, but we really wanted to tie that to bleaching observed in the field. So we know that degree heating weeks correspond to real coral cover on the Great Barrier Reef. So about 4-degree heating weeks equals about a 45% decline in coral cover. 8-degree heating weeks can equal about a 65% decline in coral cover. So we really wanted to tie what we were seeing in our communities with the heat experience on these reefs to actually how much bleaching was observed. So we reached out to Terry and he sent us the aerial bleaching scores, and we were able to calculate a residual degree heating week metric. So essentially, given how much heat accumulated on those reefs by looking at the satellite data for each of our different reefs, so given how much heat accumulated on those reefs, actually how much bleaching was recorded. So this is what was described in our residual metric of degree heating weeks. And if you plot that a different way, here we have on the x-axis the aerial bleaching score. So that score that the ARC Center of Excellence collected through their aerial surveys, where 1 is a not very bleached reef and 4 is a pretty bleached reef. And then we compared our satellite data for each of those reefs on the y-axis, and we could see that these 4 reefs that I've highlighted here, these are reefs that pretty much cooked. So that's 10-degree heating weeks. So if we think about 8 contributing to such a strong decline in coral cover, 10 is quite significant. But for some reason, these reefs did not record much bleaching. And that could be in comparison to reefs that did experience quite a lot of heat, and did bleach accordingly. So we were really interested in these 4 maroon reefs, so the toughest reefs. And so we also were interested in taking our toughest coral, Acropora tenuis, and then looking at the symbiont communities in those corals. And what we found when we juxtaposed the difference between those 2 mechanisms that I described earlier, the shuffling and the switching, is that the toughest corals at these toughest reefs did both. So they had about an even split between using these 2 mechanisms to potentially contend with this additional heat that they were experiencing. And in particular, C3K really did seem to pop up, and that's potentially a symbiont to look at in the future. So why is it important that not only we understand what's the symbiont community inside of corals after bleaching, but also free living in the environment. Well, we know that the sediments and the water column are really important reservoirs for symbionts after mass coral spawning, seen here. So on the Great Barrier Reef, about 85% of coral species actually need symbionts free living in the environment to take them up and then to establish symbiosis anew. So it's really important that we understand actually what's available to them moving forward for generations to come, because that could really help to dictate how they fare in future bleaching events. So in summary, we found that most corals did not have these stress-tolerant symbionts before 2016, and that the diversity of these this Durastinium group really exploded after this first bleaching event, and that interestingly, both coral species as well as different reefs responded really differently to the level of heat that they were exposed to. So I'd just like to say thank you, and I'll come back and we can ask questions about that afterwards. But I'll just go on now to talk a little bit about Mindaroo Foundation. So a bit changing gears a bit. So Mindaroo is a philanthropic organization, and I didn't know this a few months ago, but it's actually one of Australasia's largest philanthropies. So 2.6 billion currently invested. And essentially, we look for really tough, persistent issues with the potential to drive big change. So we're constantly looking to support researchers and activists that have game-changing ideas that would really benefit people around the world. And we also are an incubator of ideas in order to accelerate impact. So we do a number of different things, both in the support as well as in the idea generation space. And a few of the initiatives currently on the go include facilitating the elimination of childhood cancer, with some really incredible work going on in hospitals in tailored medicine in New South Wales, the ending of modern slavery, and then probably most relevant to this group, driving accountability in global overfishing, plastic pollution, and global warming. So some pretty meaty topics. And so I'll just kind of take you through our planet portfolio. So within the planet portfolio, we have flourishing oceans, and I've highlighted that because I'll talk about that next. Plastics, as well as fire and flood resilience. You've probably seen some of those Mindaroo pods being deployed to some of those flood-prone areas down in New South Wales. And most, I guess, new on the horizon is new initiatives around animal welfare. So within the flourishing oceans, we have a number of different teams. So we have the ocean conservation team, we have the sustainable fisheries team, we have research and infrastructure, we have oceanomics, and then we have marine education. And I'll kind of just be taking you through a few of these examples in the slides to come. But some of the kind of things that we've been supporting include the publication of an inaugural Global Fishing Index, now online, and I'd really invite you all to go take a look if you're into that kind of space. Currently 10 million kilometers squared of marine protected areas are currently in the process of being implemented in the ocean conservation through the Ocean Conservation Group. The Oceanomics Center build has been completed and is preparing for opening, and that's a really beautiful center centered around the University of New South Wales in Perth. And near and dear to my heart is the MIRL, or the Mindaroo Exmouth Research Laboratory, is now open and fully operational in Exmouth on the doorstep of Mindaroo Reef. So those are a few of the things that have happened in just the last few months at Mindaroo. So ocean conservation. So this project really aligns with the UN's goal, 30 by 30 goal, which is to increase the protection of the oceans to 30% by 2030. So currently only about 3% are protected. So there's really a lot to protect. And this really has to deal with acceleration of the establishment of no-take actively co-managed MPAs. And a few of the projects that are currently on the go include Project Ocean Forward, Project Mindaroo MPA Monitoring, and Project Blue Nature Solutions. And Blue Nature is a large consortia of many groups, including Mindaroo as one of the founding members. So that's the Ocean Conservation Group. Our Sustainable Fisheries Group looks at taking action to end overfishing. And this is developed in predominantly 4 different ways. So the development of sustainable small-scale fisheries, holding distant water fishing fleets accountable, improving fishing, monitoring, compliance, and surveillance, as well as ending a market access for illegally caught seafood. So not just calling out and helping to develop technologies around identifying illegal seafood, but actually pushing for change where it's most needed. So the Ocean Omics Group looks towards revolutionizing how we measure, understand, and conserve life in the ocean. And a lot of this has to do with marine genomics and enabling technology and really cutting edge technological solutions to map and monitor large areas of the ocean. And some of that work centers around eDNA, which I'm sure many of you are aware of, as well as building ship-based and autonomous platforms to enable real-time and ecosystem scale deployment of marine genomics. So people who work with genomics in this space know that it takes a long time to generate data and to go through data and to analyze it. And so trying to really bridge the gap between data acquisition and information for managers to make good management decisions. And then finally, generating and openly publishing reference genomes for a number of endangered, threatened, and unsustainably harvested species. So really trying to push for this open access, resources and tools available for all, especially in this area of creating genomes, which is a very challenging and expensive practice. And then finally, the Exmouth Research Lab, the MERL, so we call it the MERL, which is tasked with supporting and developing world-changing marine research to drive for positive change in the ocean and its communities. And so we have cutting-edge aquaria based on CSIM technology. We're situated right on Mindaroo Reef, which is a World Heritage Site in Western Australia. And you can see the beautiful Mindaroo coastline there on the slide. And we also have very sophisticated molecular testing facilities, so you can do all your lab work right there in the lab. And so that's kind of Mindaroo Flourishing Oceans in a nutshell. And I'll take any questions about the first part or the latter part as people have. Thank you.